Welcome to Tiny Expeditions, Season 7, Episode 3, the one all about peanuts.
All about peanuts, but before we start talking about peanuts again, you need to know who we are. My name is Chris Powell. I'm your co-host for this season.
And I'm Dr. Sarah Sharman, here to help you understand the science.
And Dr. Sharman, we are talking about peanuts, but specifically this season is talking about it through the lens of the WIREGRASS Peanut Project, which is a very specific application of peanut research that is happening in the Dothan area alongside of the researchers here at Hudson Alpha in Huntsville. And this project is fascinating because there's so many different elements to it, right? In our first episode, we talked about education, and we talked about how a peanut can be used to inspire and to train the next generation of scientists in our school systems now. Last episode, we talked about how peanuts can be used to drive economic growth in specific areas. In this episode, well, Sarah, we're a research institute. I feel like we need to get back to the research. So take us on.
Can do, that is my specialty. So historically, when farmers are looking to breed a new kind of peanut, they're basically just taking the two peanuts that have good traits that they're looking at and breed them together and hope for the best. So that is a long, laborious process. You have to wait until the peanuts mature in the field to see what you got. However, genetics is helping to speed that up. So scientists are looking at peanuts that farmers have deemed good, and they're digging into the DNA and really trying to connect the genetics with the traits that matter. So then you can look at the genetics of these peanuts and choose to breed them together and really get results faster. So we're talking about traits like drought tolerance, pest resistance, growing in certain types of soil. So really just lumping it all together and making the perfect peanut.
This is a very complicated process, so we need very smart people working on this. And it just so happens that we have one of the world's best peanut researchers here at the Institute, Dr. Josh Clevenger. So let's meet him now.
Yeah, my name is Josh Clevenger. I'm a faculty investigator here at Hudson Alpha Institute for Biotechnology. I focus on translational genomics, so I'm really excited about how we read DNA at scale and interpret it at scale to accelerate crop development and help feed people.
So you are known as a primary expert in the world of peanuts.
That's right, yeah.
How did you become that expert? What brought you to peanuts?
It's actually a really nice story, I think, for young people that are sort of at the early stages of their career in science or any other career, and you think about who am I going to be, who am I, what do I care about? The way I got to peanuts was really about me as a person understanding what was going to excite me about getting up out of bed every day. And it was basically working in crop improvement and breeding to help sort of develop plants that could be more sustainable. And then when I was working on tomato fruit shape, I was just so excited, over the moon about I was understanding how those genes created different shapes of tomatoes. And the question after I would give these presentations, and I felt like I was so naive, I felt so amazing about, oh, that was so great. The question was always, who cares, right? And so that really kind of hurt me deeply. And I was like, oh my gosh, like, I think this is great, but no one seems to think that it matters. And so I decided, okay, I'm going to work on something that matters. I don't want to hear who cares ever again in my entire career. And I remember thinking that, like, I'm not just saying that. I thought that I don't want to hear that ever again. And I applied to PhD programs and got basically rejected from all of them. Then my mentor, Peggy Ozias-Akins, reached out way late in the game and said, hey, it's a really funny story. She said, I got a USDA grant that it took so long to fund, I forgot I put it in. And I need a graduate student, and there's none left because they were all taken, right? And I need to start. But, like, I saw your application. Are you still interested in coming here? And I said, yes, please. But that was on aflatoxin and peanuts. So that's how it happened. And now, after sort of learning about how incredibly healthy peanuts are, I went from probably not eating them at all to now eating them every day. And going from like peanut butter and jelly is a stupid thing my mom put in my lunch to like, this is a really great food to eat every day. So it's kind of an interesting transition.
So to push a little bit further into the latter half of that, how are you using genetics to really speed up the rate at which we can develop these beneficial varieties?
You always are thinking about the perspective of the time that you're living in. And you always feel like maybe it's the best time. Because when I started science, and it wasn't that long ago, 15 years ago, the aspect of like reading the genome of hundreds of thousands of individuals was, you weren't going to be able to do that. We'd like talked about it. Oh, in the future, we'll have to know how to do that. Now, like I routinely am, sequencing peanuts, which at the beginning of my career, it took 12 years and $12 million and 100 global scientists to sequence one peanut. Wow. Now, every time we have a new like peanut of interest or parent or whatever, we sequence it. It takes a day or two. It costs $1,000. And it's very routine. And so we have hundreds and hundreds of sort of genomes, and then we can go back and actually layer on sort of reading most of the DNA of tens of thousands of individuals. Okay, so we have this ability. How do you do it? How do you use it? So one of the things that in this time is really interesting to me is that a lot of the genetics have become mature for most of the crops that we haven't used genetics on before. And there's all these incredibly high effect sort of pieces of the genome that we can identify and then use in breeding, right? So I was talking about, maybe fortifying, like increasing an amino acid or something. There, a lot of times there is a high effect locus, we call it a piece of the region of the genome where there's a variant of a gene that we can identify, and then that has a big effect. And we haven't had access to that before, so the germplasm doesn't have those things included into it. But every time you find an individual, and maybe humans are like this too, every time you find an individual that has this one amazing trait, the rest of their traits are like kind of terrible. Maybe humans aren't like that, but some are. So if you made a cross, if you think about how breeding happens, if you make a cross between a peanut that like is the best boiling peanut ever and is a high yielder and everyone likes growing it because of its attributes agronomically, but this peanut doesn't have any of those things, but it has this one thing I want to include. So if you just imagine how sort of independent segregation works, which means like the DNA completely segregates independently, you can't control it. If you make that cross, you would have to look at hundreds of thousands of individuals to find all the good things here and the good thing here. But if you could read the DNA quickly, you can immediately understand which individuals sort of have a leg up and have most of the things that you want. So what we do is we do genetics to first identify those loci, which there's a lot of them still that have high effect that we can identify. And then within our populations, we can just take little slivers of the seed and extract DNA and sequence that genome and say, this individual has a bunch of the things I really like, and it has a bunch of the things I really like over here, and so I'm going to move forward with that. Whereas before, what you'd have to do is put it out in a field, which you only had so much space and money to take care of it, and hope that one of those, I have space for 300, hope that one of those is one of the ones. And also, which I find, I always think about this, like phenotypes are hidden most of the time. Like you don't always see them. They're not always obvious. So not only do you have to get that individual, but you have to catch it. You have to capture it. You have to actually find it and not miss it. And I think that's where genetics and like genomics to me are so magical, is that it's like we don't have to worry about that anymore. We can always catch it, even if that plant looks exactly the same. So that's sort of how we do that.
And are you working on anything right now in peanut that excites you?
Oh yeah. peanut's like a little bit boring. Like it's not boring, but it's a little bit boring because it just sort of like became peanut like 10,000 years ago, which if you think about like the time it takes to evolve really fun things, that's not enough time. And so it hasn't really, it's a baby, you know? And so there's one locust that provided virus resistance. And for some reason, by chance, and these things sort of happen in genomes, which is also so fascinating. By some reason, by chance, a gene from a family that helps provide resistance to pathogens jumped to another chromosome. And then where it was, there was some signature in the DNA that made it so that it would duplicate itself. And in the process of duplicating itself, it made it superpowered and gave it this amazing resistance to this virus that nothing else has, right? Like everything else just gets crushed. But like this sort of like duplication of this gene makes a superpower. And so like that's cool. But like if you think about all the plants that are probably have done things like this, like you have to find it, right? And it has to be in like your plant of interest. So for some reason, fate put me in touch with this particular trait. And for some reason, I found it, you know, just dumb luck, a little bit of being good at stuff, but like mostly dumb luck. And it's absolutely fascinating because this gene family is so incredibly important for resistance to all pathogens. And there's only 20 members of it. I just sort of have been thinking about this. And so I'm like, wow, only 20 members. Like, it's so important, but like, it's like this like little like elite club that like just like devastates fungi and bacteria and viruses. And peanut is the little sort of orphan that got it to duplicate itself and show its superpower. And then I got to find it. And so that's really exciting because what we're doing now is we're saying, what can we learn from that? And how can we use it to like engineer new kinds of resistance? And I have this really incredible friend who works with me now. And he has he actually published on this, I don't know, 15 years ago, about like he found signatures in the DNA that like would lead to tandem duplications. So how do these things occur? And sort of he hypothesized, he did this in rice, like these signatures would allow tandem duplications. And then this German researcher, which I joke with him about, this German researcher took that paper and experimentally validated and showed that, yes, this is true. And in his follow-up paper, he called my friend a pioneer of bioinformatics. And so I'm always joking with him, calling him a pioneer of bioinformatics. But what's cool about that is that if you're following this story, you're sort of putting the pieces together. He has a hypothesis of how to create tandem duplications. We have this gene family which we've seen this superpower where they duplicate, they get this power. There's 20 of them. So now we're thinking about creating different sort of recipes of different family members, putting it in there so they duplicate themselves and letting it sort of like create these superpowers on its own. And like, so that's really fascinating to me as like a tool to engineer new resistance.
What has been the most impactful part of the project for you personally?
Well, there's there's multiple facets of that. One is we have made amazing progress. I mean, we're putting out our first replicated yield trial with lines that have, 3 and 4 disease resistances and are probably high yielding and just really, really cool. Like the progress that we've made and the buy-in and the excitement of the community in terms of like, really like being behind it and feeling like, scientifically someone that works on breeding or works out in the field, they get this label applied. You do applied science. And in general, it's a little bit like, looking down on you, you're what you do is kind of like, we need it, but, it's not as good as what I do. I do basic science, So normally you sort of work in this, it's really like like self sort of propelled, you're not getting external validation. You're not getting applauded for the things you're doing. You're doing it because you want to do it. And to have like a whole community, like kind of like get stoked about peanut breeding, that's pretty rad, because like I didn't expect that, right? Like I knew that we would do it and there was buy-in from like the leadership and they understood what we were trying to do. But like, man, I go down there and I'm at the Peanut Fest and like people are like, "Oh, Dr. Clevenger, what's up?" Like, they want to ask about it. They want to talk about it and they're excited about it. And that's pretty cool. But I think they should be because, I don't think there's anything like this anywhere else where like a whole community participates in a process to like enrich themselves, right? Like if we succeed, like we might fail, which in science you have to accept that early on because you're going to fail most of the time. We might fail, but if we succeed, like we all did it together and like there's going to be big gains for everybody, and that's pretty cool.
Dr. Clevenger's team is really making great progress on the next great wiregrass peanut. And when you picture his lab, you might think of a lot of scientists working at lab benches, doing actual experiments with peanut seeds and all that. And they do, but not everybody in his lab is a bench scientist. A lot of his lab actually works on computers, going through all of the data that they're generating. We sat down with one of those computational biologists who works in the wiregrass, Catherine Davis.
My name is Catherine Davis, and I live in Dothan, Alabama with my husband, 3 dogs, 3 kids. And my area of expertise is computational biology, and I work for the Clevenger Lab. So my main focus is to process all of the Wiregrass Peanut Project data.
So computational biology is a newer field. For our listeners who don't know what that is, can you explain what computational biology is or how you're using it for your current job?
Yeah, so the genomes that we're working with are really large. Like the peanut genome is about 2.7 gigabases, so that's like 2.7 billion base pairs. And it's, you're just looking at letters. When you look at it on a screen, if you printed it all out, it's just 2.7 billion letters. And how do we make sense of that? And how do we make sense of the data that we're sequencing from the peanut plants? And we have tools that have been developed by other people and also tools that our computational team has kind of tailored to the data that we work with to be able to generate patterns and insight from those data sets. And that's kind of what computational biology is. It's making sense of all of the work that the wet lab has done, and then kind of transforming it into a format that we can actually make decisions with.
So when you're looking through this peanut data set, what are the questions and problems y'all are trying to solve within the Wiregrass project?
The main focus is to improve peanuts, right? So being able to find plants that have the gene of interest. And so, that might involve comparing ones that we do see have that versus ones that don't, and making sure that we're seeing that consistent change in whatever that region of the genome is, and saying, okay, looks like this plant, has that trait that we want, and trying to incorporate that so we can make a better peanut.
In the wiregrass, most people know this area for peanuts, and they think of farming when thinking of this area. And you were living here before you started working with Hudson Alpha Wiregrass,
Correct.
Right? So did you find that there's actually an abundant offering of jobs for your skill set here?
Absolutely not.
No. Okay. So now there is an option for you to have that job here at your home. So can you talk to us about that? Like what is it like to be able to specialize in this particular field and you're able to actually like go to work at home, like in this area? And what does this mean to you to have this project here in your backyard?
So I had mentioned earlier that, I had flip-flopped a lot about what direction I was going to go with biology. And there aren't very many opportunities like this down here. This is the first one that's even close to being in this realm of things. So that was really exciting for me, because it was important to me that whatever I'm doing, it feels meaningful and it makes an impact, and this does, and I see that every day. So to have this, in our own backyard is a huge deal. It's a huge deal for Dothan. This is the first of its kind here, and I know from people talking around town, like they're really excited about it. They're asking all the time, like, what is the Wiregrass Project? What are we doing with these peanuts? I keep hearing of Hudson Alpha. What is Hudson Alpha? Where are y'all at? And so yeah, it's exciting.
Hopefully you've enjoyed these 2 interviews with Dr. Clevenger and also with Catherine. Their stories are fascinating, Dr. Sharman. I mean, just listening to where they've come from professionally, to see the journey that got them to where they are, the research that they're working on, but specifically the real-world impact that's happening because of what they're doing. So before we go any further, though, let's kind of wrap things up and let's see, where have we gone over this episode? What stuck out to you? Let's do a TLDR.
Sure, TLDR. I think for me, I would be remiss if I did not talk about the research, but really just the true breadth of important things we can solve with DNA. So obviously this episode was all about how we can improve the peanut by looking at the peanut's DNA, but that's applicable to really everything across human health, agriculture. So just really how cool DNA is, I guess is my answer.
Okay, so that's a great one. And can I go alongside of that? There was something that we're leaving out of this episode that we're going to put in as bonus content, but it was a really great story. And you need to go look this up. But Dr. Clevenger told us a couple of different stories about how peanuts are being used to transform societies around the world. He gave us one example of a story from Asia, one from, I believe it was in Ghana. And the one in Ghana really stuck out to me. And again, go look at it. But the whole point of the story is using peanuts through peanut butter to supply nutritional value for children who are needing surgeries. And there's a lot more to the story. Go look it up. It was a fascinating story. But to think that this research is being tied to children receiving surgeries in Ghana to be able to be accepted by communities. Fascinating connection.
Thanks for joining us for this deep dive into the research behind the Wiregrass Peanut Project.
Join us for the next episode where we will follow one peanut plant's journey from the classroom out into the field.
Tiny Expeditions is a podcast about genetics, DNA, and inheritance from the Hudson Alpha Institute for Biotechnology. We're a nonprofit research institution in Huntsville, Alabama, with a very unique mission.
We bring together scientists and companies to develop and apply genomic advances to make a better world. And that includes everything from brain research to agriculture for a changing climate.
If you enjoyed this episode, please swing by your favorite podcast app and hit that subscribe button. While you're there, consider leaving us a review. It really helps us spread the knowledge. Thanks for joining us.